This project began as an art project exploring the relationship in parasitism. From 2019 to 2023, this research evolved through collaboration between artists, scientists, and engineers, with scientific theory and data transformed into a simulation of the interaction between viruses and human molecules.
COVID-19 has disrupted our healthcare, social, and economic environments. In spite of significant technological advancements made in managing this pandemic over the last 3 years, a substantial number of people are still infected and suffering from the virus. This project aims to focus on the mutualistic relationship between parasites and humans by referencing scholars in contemporary philosophy and bioscience domains such as Richard Dawkins, Lynn Margulis, and Donna Haraway. In this sense, parasitism has been studied in this series of works as an interspecies relationship between humans and other species, examining various contexts in social, artistic, and scientific practices. Therefore, our research question aims to provide new perspectives on scientific discoveries in complex biosystems by creating interactive audiovisual simulations that depict the physical characteristics of the interactions between the coronavirus and human protein molecules. This approach not only aims to impart educational knowledge, but also to encourage conversations from diverse viewpoints encompassing art, science, and technology.
This project started with a scientific observation of the interaction between the SARS-CoV-2 virus and the human cell components. At UNS Kepler University Linz here, we operated high-speed atomic force microscopy and successfully recorded the first movie of the trimer of the SARS-CoV-2 virus. This breakthrough allowed us to characterize how the coronavirus can interact with a human protein inside the human body.
A custom instance rendering method is implemented in AlloLib, which is open source C++ platform developed for immersive interactive media, using OpenGL shading language to draw the dynamic and volumetric structure of the virus spikes. By filling out hundreds of thousands of vertices with transparent instances, the skeleton of the 3D model closely resembles the original AFM image. We brought the infection model of coronavirus from the conducting zone of the airway to cellular membrane with ACE2 receptors. This project's narrative is driven by the infection mechanism comprised of the appearance, diffusion, penetration, and binding for entry. Just like the actual infection progress, these stages can go back and forth or be accelerated or decelerated by the probability model and the real-time interactive components.
We designed the real-time sonic landscape to sonify the data and the underlying process of infection, but also to compose a musical plot structure that aligns with the events in the simulation. As a result, the sonic landscape is filled with unique and dynamic patterns that are interconnected with the simulation, visualization, and narrative.
We designed the interface to invite the audience to participate in the audiovisual narrative. The interface incorporates two methods, a voice-based interface and a touch-based interface, allowing users to intuitively explore the dynamic biophysics of the coronavirus and human cells.